Acinetobacter, Wikipedia article audio. Acinetobacter albensis, Acinetobacter apis, Acinetobacter baumannii, Acinetobacter bigerunckii, Acinetobacter berezania, Acinetobacter bohemicus, Acinetobacter boissieri, Acinetobacter bovitii, Acinetobacter brissui, Acinetobacter chalcoacidicus, Acinetobacter corvallinii, Acinetobacter dispersus, Acinetobacter equi, Acinetobacter gandensis, Acinetobacter jerneri, Acinetobacter guangdongensis, Acinetobacter gillorii, Acinetobacter gillenbergii, Acinetobacter hemolyticus, Acinetobacter harbinensis, Acinetobacter indicus, Acinetobacter junii, Acinetobacter cooki, Acinetobacter olwoffii, Acinetobacter modestus, Acinetobacter nectaris, Acinetobacter nosocomialis, Acinetobacter parvus, Acinetobacter pakistanensis, Acinetobacter populi, Acinetobacter proteolyticus, Acinetobacter pittii, Acinetobacter puyangensis, Acinetobacter kingfengensis, Acinetobacter radioresistens, Acinetobacter rudis, Acinetobacter skindleri, Acinetobacter seiferdii, Acinetobacter soli, Acinetobacter tandotu, Acinetobacter tetrunbergii, Acinetobacter towneri, Acinetobacter ursingii, Acinetobacter variabilis, Acinetobacter venetianus, Acinetobacter vivianii. Acinetobacter is a genus of gram-negative bacteria belonging to the wider class of Gamma-proteobacteria. Acinetobacter species are oxidase-negative, exhibit twitching motility, and occur in pairs under magnification. Description. Etymology. They are important soil organisms, where they contribute to the mineralization of, for example, aromatic compounds. Acinetobacter species are a key source of infection in debilitated patients in the hospital, in particular the species Acinetobacter baumannii. Species of the genus Acinetobacter are strictly aerobic, non-fermentative, gram-negative bacilli. They show mostly a cocobacillary morphology on non-selective agar. Rods predominate in fluid media, especially during early growth. The morphology of Acinetobacter species can be quite variable in gram-stained human clinical specimens, and cannot be used to differentiate Acinetobacter from other common causes of infection. Most strains of Acinetobacter, except some of the Olwoffii strain, grow well on MacConkey agar. Although officially classified as not lactose fermenting, they are often partially lactose fermenting when grown on MacConkey agar. They are oxidase negative, catalase positive, indole negative, non-modal, and usually nitrate negative. Bacteria of the genus Acinetobacter are known to form intracellular inclusions of polyhydroxyechinotes under certain environmental conditions. Taxonomy. Acinetobacter is a compound word from scientific Greek, meaning non-modal rod. The first element acineto appears as a somewhat Baroque rendering of the Greek morpheme alpha kappa nu eta tau omicron dash, commonly transliterated in English as acinetto, but actually stems from the French syntaxe and was adopted directly into English. The genus Acinetobacter comprises 38 validly named species. Identification. Identification of Acinetobacter species is complicated by lack of standard identification techniques. Initially, identification was based on phenotypic characteristics such as growth temperature, colony morphology, growth medium, carbon sources, gelatin hydrolysis, glucose fermentation, among others. This method allowed identification of Acalcoacidicus A. baumannii complex by the formation of smooth, rounded, mucoid colonies at 37 degrees Celsius. Closely related species could not be differentiated, and individual species such as A. baumannii and Acinetobacter genomic species 3 could not be positively identified phenotypically. Because routine identification in the clinical microbiology laboratory is not yet possible, Acinetobacter isolates are divided and grouped into 3 main complexes. Different species of bacteria in this genus can be identified using fluorescence lactose denitrification to find the amount of acid produced by metabolism of glucose. The other reliable identification test at genus level is chromosomal DNA transformation assay. In this assay, a naturally competent tryptophan oxotrophic mutant of Acinetobacter bailii is transformed with the total DNA of a putative Acinetobacter isolate, and the transformation mixture is plated on a brain-heart infusion agar. The growth is then harvested after incubation for 24h at 30 degrees Celsius, plating on an Acinetobacter minimal agar, and incubating at 30 degrees Celsius for 108h. Growth on the AMA indicates a positive transformation assay and confirms the isolate as a member of the genus Acinetobacter. E. coli HB101 and Acalcoacidicus MTCC1921T can be used as the negative and positive controls, respectively. Habitat. Some of the molecular methods used in species identification are repetitive extragenic palindromic sequence-based PCR, ribbity-ping, pulsed field gel electrophoresis, random amplified polymorphic DNA, amplified fragment length polymorphism, restriction, and sequence analysis of tRNA and 16S to 23s rRNA gene spacers, and amplified 16S ribosomal DNA restriction analysis. PFGE, AFLP, and ARDRA are validated common methods in use today because of their discriminative ability. However, most recent methods include multi-locus sequence typing and multi-locus PCR and electrospray ionization mass spectrometry, which are based on amplification of highly conserved housekeeping genes and can be used to study the genetic relatedness between different isolates. Clinical significance. Acinetobacter species are widely distributed in nature, and commonly occur in soil and water. Their ability to survive on moist and dry surfaces, as well as to survive exposure to various common disinfectants, allows some Acinetobacter species to survive in a hospital environment. Furthermore, Acinetobacter species can grow at a broad range of temperatures, allowing them to survive in a broad array of environments. Treatment. Acinetobacter is frequently isolated in nosocomial infections, and is especially prevalent in intensive care units, where both sporadic cases and epidemic and endemic occurrences are common. A. baumannii is a frequent cause of hospital-acquired pneumonia, especially of late-onset, ventilator-associated pneumonia. It can cause various other infections, including skin and wound infections, bacteremia, and meningitis, but A. lwophi is mostly responsible for the latter. Of the Acinetobacter, A. baumannii is the greatest cause of human disease, having been implicated in a number of hospital-acquired infections such as bacteremia, urinary tract infections, secondary meningitis, infective endocarditis, and wound and burn infections. In particular, A. baumannii is frequently isolated as the cause of hospital-acquired pneumonia among patients admitted to the intensive care unit. Risk factors include long-term intubation and tracheal or lung aspiration. In most cases of ventilator-associated pneumonia, the equipment used for artificial ventilation such as endotracheal tubes or bronchoscopes serve as the source of infection and result in the colonization of the lower respiratory tract by A. baumannii. In some cases, the bacteria can go on to enter the bloodstream, resulting in bacteremia with mortality rates ranging from 32% to 52%. UTIs caused by A. baumannii appear to be associated with continuous catheterization, as well as antibiotic therapy. A. baumannii has also been reported to infect skin and soft tissue in traumatic injuries and post-surgical wounds. A. baumannii commonly infects burns and may result in complications owing to difficulty in treatment and eradication. Though less common, some evidence also links this bacterium to meningitis, most often following invasive surgery, and in very rare cases, to community-acquired primary meningitis wherein the majority of the victims were children. Case reports also link A. baumannii to endocarditis, keratitis, peritonitis, and very rarely fatal neonatal sepsis. Aseptic technique. The clinical significance of A. baumannii is partially due to its capacity to develop resistance against many available antibiotics. Reports indicate that it possesses resistance against broad-spectrum cephalosporins, beta-lactam antibiotics, aminoglycosides, and quinolones. Resistance to carbapenems is also being increasingly reported. A. baumannii can survive on the human skin or dry surfaces for weeks, and is resistant to a variety of disinfectants, making it particularly easy to spread in a hospital setting. Antibiotic resistance genes are often plasmid-borne, and plasmids present in Acinetobacter strains can be transferred to other pathogenic bacteria by horizontal gene transfer. In healthy individuals, Acinetobacter colonies on the skin correlate with low incidence of allergies. Acinetobacter is thought to be allergy protective. Acinetobacter species are innately resistant to many classes of antibiotics, including penicillin, chloramphenicol, and often aminoglycosides. Resistance to fluoroquinolones has been reported during therapy, which has also resulted in increased resistance to other drug classes mediated through active drug efflux. A dramatic increase in antibiotic resistance in Acinetobacter strains has been reported by the Centers for Disease Control and Prevention, and the carbapenems are recognized as the gold standard and treatment of last resort. Acinetobacter species are unusual in that they are sensitive to sulbactam, which is commonly used to inhibit bacterial beta-lactamase, but this is an example of the antibacterial property of sulbactam itself. In November 2004, the CDC reported an increasing number of A. baumannii bloodstream infections in patients at military medical facilities in which service members injured in the Iraq/Kuwait region during Operation Iraqi Freedom and in Afghanistan during Operation Enduring Freedom were treated. Most of these were multi-drug resistant. Among one set of isolates from Walter Reed Army Medical Center, 13 were susceptible to imipenem only, and 2 were resistant to all drugs tested. One antimicrobial agent, colistin, has been used to treat infections with multi-drug resistant A. baumannii. However, antimicrobial susceptibility testing for colistin was not performed on isolates described in this report. Because A. baumannii can survive on dry surfaces up to 20 days, they pose a high risk of spread and contamination in hospitals, potentially putting immunocompromised and other patients at risk for drug-resistant infections that are often fatal and, in general, expensive to treat. Reports suggest this bacterium is susceptible to phage therapy. Natural transformation. Gene-silencing antisense oligomers in a form called peptide-conjugated phosphorodiamidate morpholino oligomers have also been reported to inhibit growth in tests carried out in animals infected with antibiotic-resistant Abomanii. The frequency of nosocomial infections in British hospitals prompted the National Health Service to research the effectiveness of anions for air purification, finding that repeated airborne Acinetobacter infections in a ward were eliminated by the installation of a negative air ionizer. The infection rate fell to 0. Acinetobacter chalcoacidicus bominii complex, glucose-oxidizing non-hemolytic, Acinetobacter olwofii, glucose-negative non-hemolytic, Acinetobacter hemolyticus, hemolytic. Bacterial transformation involves the transfer of DNA from a donor to a recipient bacterium through the intervening liquid medium. Recipient bacteria must first enter a special physiological state termed competence to receive donor DNA. A chalcoacidicus is induced to become competent for natural transformation by dilution of a stationary culture into fresh nutrient medium. Competence is gradually lost during prolonged exponential growth and for a period after entrance into the stationary state. The DNA taken up may be used to repair DNA damage or as a means to exchange genetic information by horizontal gene transfer. Natural transformation in a chalcoacidicus may protect against exposure to DNA-damaging conditions in the natural environment of these bacteria, as appears to be the case for other bacterial species capable of transformation.